Hello everyone. My name is Mladen, Senicar, Research Associate here at ACD's Pharma Assay Services. And yeah, let me just skip to the first slide. So as I said, my name is Mladen Senicar, Research Associate here at ACD's Pharma Assay Services. I'm joined here today by my colleague, Jyoti Sheldon. She's our Associate Scientist from our Applications group. And before we get started, I just want to I just hope you and your loved ones are staying safe and healthy in these unprecedented times. And just want to let you know that we at Bio-Techne's advanced cell diagnostics are considered an essential business. Therefore, we are fully operational and open to helping our collaborators with their immediate needs. All right, so today's presentation, as Ron already mentioned, is going to be about viral detection with ACD's in situ hybridization assay services. So with that said, this is our agenda for today. And we just want to let you know before we get started that Pharma Assay Services PS is the only CRO specialized in providing RNAISH services and can help you visualize and quantify target gene of interest with morphological context as well. We'll dive deep into that later on. This webinar, as you can see, based on the agenda, we'll introduce our RNA Scope and base scope in situ hybridization-ish services, our capabilities and workflows, and use of our quality control tissue bank for target validation as well. So then we'll move on to how ACD's assay service offerings can be leveraged to detect viral infections and pathogenesis and enhance our understanding of global important viruses such as SARS-CoV-2. Also, we'll be discussing the detection of AAV and LV vector biodistribution and transgene expression in preclinical animal models with in situ hybridization technology as well. So we'll walk you through all of these, and then we'll wrap this up with some interesting viral detection case studies, and we'll talk a little bit more about the trending viruses and how they can be detected by our RNA Scope-ish analyses. Okay, so when we get started, let's see what is RNA Scope and how can we use that to combine single cell transcriptomics with spatial analyses. So in this first slide, we're trying to do a little comparison, a little analogy. So traditionally, transcriptomic analysis have relied on bulk tissue analyses such as PCR, which we all know and are pretty familiar with. This, for example, PCR provides an average level of gene expression in a sample, but loses the spatial and single cell information. So this is pretty analogous to a fruit smoothie, which can be a delicious drink, but you can only guess that if you can only guess what's in the smoothie. So you don't really know the ingredients. Moving on, we also have single cell RNA sequencing that has shown that we can use that platform for knowing researching molecular information at a single cell level. And to come back to the fruit analogy, we cannot provide you with a menu and tell you what fruit and how much of each fruit went into that smoothie. However, these techniques still do not provide you with how those cells are organized in the tissue context. So with that being said, this is our way of kind of combining best of both worlds, where we can see single cell spatial analyses by RNA Scope-ish assays. So this is where our spatial analysis comes into play, and then single cell multiplexing spatial analyses that detects RNA can provide both transcriptomic information with spatial organization while retaining the single cell resolution as well. So as I mentioned earlier, this combines best of both worlds, so to speak. So now going back to the fruit analogy, as you can see on the far right side, instead of an average blended smoothie with mystery ingredients, you can see all the fruit and how it is spatially arranged. Okay, so moving on, this is just going to be a little sneak peek into AAV and transgene detection that we'll talk more in depth later and show some cool and interesting case studies. So as you will observe here, RNA Scope technology is being leveraged for detection of AAV-based viral DNA and transgene RNA detection. And on the right side, you guys can see in eye tissue, in liver tissue, and also AAV transduced brain tissue as well. So we'll go back to this slide later on. Moving on to the next slide, this is another representation of our quick sneak peek, so to speak. And then you guys can see how these virally induced tissues look under a microscope or under a scan imaging. Okay, so to kind of jumpstart this RNA Scope overview and over the whole technology, we're gonna do a little overview of the RNA Scope technology, as I already mentioned. So how are we able to do the previously virally transduced detection using virally transduced tissue and detect these transgenes using RNA Scope. So the RNA Scope technology is an ideal spatial analysis solution to interrogate complex tissues and is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single cell level. So as you can see, excuse me, in this slide, it consists of 3 parts. The first part you see on the far left is a unique target probe that ACD designs against your sequence of interest and and then a single amplification system that generates a high signal to noise ratio. This is if we're moving from left to right, so signal amplification part. And then lastly, visualization of that set signal in representation of punctuated dots, which are our RNA transcripts or our RNA molecules. So this assay allows for the spatial mapping of a variety of different applications for mapping of messenger RNA, long non-coding RNAs, splice variants, point mutations, highly homologous sequences in cells and intact tissues as well. These all can be visualized, as you can see on the right side, which we'll dive a little bit deeper later on. Can be visualized with fluorescent and chromogenic labels. Okay, so this assay can be also performed on a variety of sample types, including, so we kind of work with multiple different tissues, including FFPE tissues, fresh frozen or fixed frozen tissues, PBMCs, and also cultured cells. Okay, so the first part of RNA technology is, of course, the probe design. So what's a Z? Z definitely, that's what we used to refer to our probes. Z's are actually a combination of 3 different things. So the bottom line would be our target specific binding site, which binds to the actual gene of interest. The middle part is a linker between the top and bottom, and the top part is our pre-amplifier binding site. So the key feature of our RNA Scope technology is the probe design I already mentioned. So we depict the oligonucleotide target-specific probes as Z's to emphasize the fact that we have 2 regions linked by a spacer, which I've already briefly mentioned, which is pretty well depicted on the slide. So when 2 Z's hybridize in tandem to the target sequence, it creates a unique binding site upon which pre-amplifiers can bind on the amplification tree and then build the amplification tree, which we'll see in the upcoming slides. So moving on, a standard RNA Scope probe for targeting sequences of 1,000 bases or more will consist of around 20 ZZ pairs. So as you can see, each ZZ pair can bind to around 50 base pairs. So that would add up to around 1,000 bases overall per probe. They hybridize next to each other and then along the target region as well. This allows for a tremendous amount of application and signal potential. However, only a few ZZ pairs are enough to bind and create that signal. The rest of it is mostly for specificity. Okay, so diving a little deeper into the probe sensitivity and specificity, this is what I was telling you guys before. So this diagram on the left side depicts beautifully how linker sits in between pre-amplifier binding site and the target binding site as well. So moving on, you guys can see that pre-amplifier binds to the top of the ZZ pairs, and then it binds the amplifiers as well. So we have our stem right here, and then what we like to call it in layman's terms are tree branches, which are our amplifiers. So these amplifiers are sequentially hybridizing to assemble a branching complex at each ZZ binding site. So each ZZ pair has one of these combinations with pre-amplifier and an amplifier. So this generates a visible signal after a chromogenic reaction, such as with DAB. So we have our label probes bind to our amplifiers, which are chromogenic or fluorophore-based label probes. The signal amplification strategy allows us for visualization of target RNA as a single dot, where each dot represents an individual RNA molecule, which I've already mentioned before, and the specificity of our assay relies on this ZZ pair. So on the right side, you guys can see that Z's would bind to the target sequence with application tree this would not be able to form if only one single Z is bound to the target probe. And then if that occurs, our stringency wash steps in between the hybridization steps would wash the whole probe away, and you wouldn't see any signal. So this really covers the specificity part pretty well. Okay, so with that being said, we can move on to our experimental configuration. Excuse me. So with our experimental configuration, before we start staining for the marker, we go to our quality control process as well with negative and positive control. So we have here as an example a serial section from a human lung cancer sample in which we performed a manual red chromogenic assay. And on the left side of the panel, you guys can see a section that's stained with our negative control probe, DAB, which is our sole bacterium gene that is not expressed in intact tissues. As observed in this sample, there should be no signal for DAB, ensuring that there's no nonspecific binding, or there's no background, and we have a nice signal to noise ratio as well. And this also allows us to see how the morphology looks in general without any RNA molecules being detected. So in the middle panel, you guys can see a sample stain with our positive control probe, which is our house-speaking constitutively expressed DPID or cyclophilin B, expressed in pretty much all the cell types. And as observed in this sample, you want to see a fairly uniform detection with a positive control probe, indicating good RNA quality. And then certain tissues may express this positive control probe at higher or lower level, but in general, you want to see the expression throughout the whole sample. And we set a bar where we can qualify this sample as a quality control pass. So with the results from these two probes, we can kind of slowly go into and surely go into our test probe, which in this case was PDL1 probe, which is our programmed checkpoint marker programmed death ligand 1. And then in this human lung cancer sample, we observe a strong punctuated dots with PDL1, indicating expression of PDL1 in this tumor sample. So first, we kind of qualified the sample and the tissue type. Then we moved on to the marker and saw a great deal of expression of PDL1, which was as expected. So this is pretty much in a nutshell how our experimental configuration looks like. Okay, so with that being said, we have two unique assays for spatial mapping of gene expression. So I've just gone through the RNA scope portion of it, and then we can compare that to our base scope, which again, just to reiterate, I said that the base scope targets pretty much enzyme junctions, splice variants, short, highly homologous sequences, void mutations, etc. So comparing it to RNA scope, base scope is only available in chromogenic, not fluorescent. And then singleplex chromogenic is available in automated and manual services. And then duplex chromogenic is only available in manual. And you guys can see nice images. So the top image on the right side, base scope is a singleplex. Then the bottom image is our base scope duplex done with manual staining. All right, so our newest addition to Rna Scope technology that we're slowly commercially rolling out this month, but it's been available for early access for the past couple of months, which has shown great results. It's our microRNA Scope Assay, which we use for the detection of microRNAs, siRNAs, ASOs, or our antisense oligonucleotides in tissues with spatial and morphological context at single cell resolution as well. So this is just one of our examples in the middle panel where you can see with aggregate control, there's definitely no siRNAs being detected. In the middle one, if you're targeting the specific, for the proprietary purposes, number one siRNA, you'll see detection of that one. But if you use the same probe to detect a different siRNA, you definitely won't see anything. So this is a nice little example that shows that we can also do, and that we're capable of doing the microRNA scope as well. MicroRNA Scope Assay is supported for, again, FFP, fresh frozen, fixed frozen, and cultured cells tissue types. And this is only available in singleplex for both manual and automated. And then we can do the chromogenic visualization, as you can see in this example right here. And the one cool thing about this assay is that it's compatible with ISH IHC, meaning it's compatible with co-detection with RNA, siRNA co-detection with the protein as well. So just to segue into the next part, as you know, I already mentioned ISH IHC. So this is one of our more specialized RNA scope ISH plus IHC/IF detection or assays available. So for this one, we can help further characterize cellular gene expression with combination of ISH with IHC. On the same slide, to simultaneously detect RNA and protein. So protein co-detection is a big deal for this one. So this is referred to as dual ISH IHC and can be used to characterize cell type specific expression, identify the origin of secreted proteins, visualize cell surface markers with RNA of interest, visualize RNA binding proteins and their target RNA, and also can dissect regulation of gene expression as well. Because these two are pretty much compatible with each other because they have similar workflows. And then once you've finished ISH amplification, as you can see in this top diagram right here, you can also add the IHC portion on top of that and the binding of antibodies. So yeah, I think this is our last RNA-ish, RNA scope-ish or ish IHC technology that we wanted to kind of give you an overview of. And then now we'll move on to our team, Pharma Assay Services, and what we have to offer as a group. Okay, so moving on. Okay, so this is like a brief overview of our PS workflow or Pharma Assay Services workflow, and we have a group of experts on histopathology services, and then you guys can use that for your research needs or any other biopharma or academic client or customer can use our group for any type of research needs. Okay, so diving a little bit into our overview of our workflow. So as you will see in the next few slides, we can help with accusation of tissue samples to our trusted chain of tissue providers and pre-qualification of these tissues for RNA Scope and base scope itch assays. We also work with our third-party CROs, and we can easily manage the embedding and sectioning of samples in our studies. And of course, we can use our unique experts to stain RNA Scope or base scope assays. After which we can provide quantitative assessment and provide a summary of our findings in a project report. So just to give you an example of our our client-facing PS overview. So we have a team of 20+ experienced lab staff scientists that perform RNA Scope and base scope assays on a daily basis, and then we really provide you with a wealth of knowledge of terms of our ability to troubleshoot and get the best data possible. So what we trying to strive for is the quality data with a quick turnaround time. That's basically our motto here at PS. So we service 20 of the top biopharma companies in the world. As you can see in this slide, there's a couple of examples. We generally stain more than 20,000 slides per year with with customers from over 20 different countries. So our team includes 2 pathologists as well. And one of them is a board certified pathologist. So they can perform specialized image analyses. And then we also have our Halo team, which specializes in Halo image analyses, which we'll get to a little later. And they're also available for consultations on your studies as needed when it comes to the analysis portion. Okay, so our team also works on enabling drug discovery. And you guys can see all the phases here. So pretty much what we do in terms of drug discovery, we can go all the way from target screening and expression analyses through efficiency screening, toxicology and safety, animal model studies, and then all the way to phase 1 clinical trials. So before mentioned safety and toxicology tissue screening, we have tissue microarrays. We provide tissue microarrays for highly sensitivity screening of your targets for preclinical and safety assessment. We also have curated a collection of dozens of pre-sourced, validated, and unvalidated tissue blocks. So these are all available for our PS customers, basically, to support the development of ISH probes and assays. So our tissue bank pretty much consists of a variety of different tissues and tissue types. So just to mention a few, we have a wide variety of normal and cancerous tumor-recruiting tissues, and then we have a complete set of homologous tissues with replicates from both males and females. And then we have our multi-organ tissue arrays for mouse and rat. We have HPV controls. We have HeLa cells. We outsource those as well. And then we have induced PBMC pellets as well. So pretty much this, pretty much this is what we offer in terms of our safety and toxicology tissue screening, and this is all that we can pretty much outsource, and you can find it in-house at ACD. So all these tissues can be used as directly in your PS study as the pre-treatment, or as I already mentioned, they're pre-qualified, so pre-treatment for these tissues specifically are predetermined. So the project itself and study moves way quicker with 100% verified quality of the tissue itself. And this kind of, as I said, I'm just going to reiterate. So this removes the need for QC and untested tissues, and then again, it shortens turnaround time and reduces the cost as well. Okay, so moving on to more early feasibility studies and assay development. This is also one of the key study types or project types that we do. So we have procedures that begins with the assay prototype establishment, followed by a precision and reproducibility study tested over multiple instruments to ensure the robustness and precision of the assay. And then once the images have been acquired and based on discussion with the sponsor, we establish a pathologist guide and scoring criteria. So we work with the wherever this assay development is being developed, we work with whether it's a CLIA lab or different type of lab. We'll work to see if our results align, basically. So this method development is documented in shared SOP, and study report, which includes optimized assay conditions, is also provided. So we work with our field associate scientists to transfer these types of studies to, for example, CLIA sites and CLIA labs, and then we make sure their results match, our results match theirs, pretty much so. In 2016, we collaborated with Leica to develop such CDX assays, and on the right side here on this slide, you guys can see that Rna Scope, compared to the other technologies, kind of like encompasses pretty much all of these 4 types, including morphological context, sensitivity, specificity, and tissue requirements. Okay, so moving on to the capabilities and workflow of our team. This is a pretty wordy chart, but you guys can get the point from the get-go to the reporting, and then we'll kind of go into what we provide point by point. So our project can be designed a la carte to help with overflow needs at any step. So standing only, analysis only. So each part of the study can be done separately as a one study, and then we have our ancillary offerings, including our tissues that we provide from our tissue bank, as we already mentioned. And then we also provide FFP embedding, H&E stains, and full slide scans, if that's the only thing you need, but it's also a part of a bigger project for sure. And then the assay types, as already mentioned, are RNA Scope assay, base scope assay, chromogenic assay, multiple fluorescent assay up to 4-plex, so you can target for genes at once, and then ISH-IC or ISH-IF assay, and then I'm gonna throw in the microRNA scope assay as well. And then as far as data analysis go, we have our tissue QC that we can do just separately, just that, or we can incorporate that into a bigger project. We have visual scoring, digital image analyses, which is done by our Halo team. And then we have our pathologist review, whether it's H scoring or visual H scoring. And then our pathologists can also do our H&E assessment as well. So for reporting purposes, we have study brief, project reports, and our clinical assay transfer, if it's the preclinical / clinical study. And then our study brief kind of like gives you a brief overview of what happened with the project and how the project was executed with the analyses, and the project report gives you a little bit more as far as methods and materials. Okay, so as far as our quality systems and security, here at ACD, we work under umbrella of GCLP and are currently working towards having a full GCLP certification. Our standard operating procedures govern pretty much our PS workflow, and including staining procedures, sample tracking, data management, data QC. All these things ensure that you get the best data in your hands. So as I mentioned, our quality data is of big importance here. So our regulated products are ISO certified, and then our HeLa cells, which you can see on the left side here, is a control run with positive and negative controls with every assay, so we can ensure that run is suitable. Run is the run is precise and there's no technical errors on our end. And then if HeLas don't pass, we will repeat those experiments at our own cost as well. So with respect to security, our files are shared using the secure file sharing system called Ignite, and there's a restricted access to this data. Okay, so just for the last thing for this part of the presentation, we want to go through our PS workflow. So phase one of each project, pretty much, if it's not split into different phases, if you go to phase one, phase two, and phase three, this is what you're going to start with, what we start with. So phase one is our QC for tissue quality and then QC for RNA quality, which you guys have already seen. And then we do a pre-treatment optimization as well to determine the best signal to noise ratio. And then again, our internal controls are included with each run to ensure that the maximum sensitivity is achieved. So after tissues have been qualified, RNA quality is good, no background, no specific binding, morphology is intact. We go into phase two where marker staining and whole slide scanning take part. And then this is followed by our analyses, which is our visual scoring, image analyses, reporting, and then we tend to do teleconferences with our clients and then explain the data a little bit more in depth. And if there's any issues, we can also address those on the calls. Okay, with that being said, we can go into data analyses methodologies. So this is pretty much how we analyze our data and pretty much go a little bit more in depth of what I've mentioned earlier. So one type of R, if you see the top left corner, is our visual semi-quantitative scoring, which is performed by highly trained experts. And then this is a rapid scoring with short turnaround time and speeds delivery of results and provides results at high, medium, and low levels. The other methodology we commonly use is our Pathologist H Scoring, which you can see on the right side, top right side. Pathologist H scoring is useful for marker expression patterns, and it's usually useful for heterogeneous patterns. So this method is more quantitative, involves grouping of cells into 5 bins based on the number of dots per cell, and each sample is evaluated for the percentage of cells in each bin. So this H scoring uses weighted formula, which ranges from 0 to 400. That's the number that you get towards the end, and that kind of estimates the quality or the abundance of expression for each gene of interest. So all of these methodologies are under the manual scoring umbrella. So for digital image analyses, we use HeLa software from the Indica labs. And our HeLa analysis is done by our team, high qualified image analysts. With HeLa analysis, we provide expression data on cell-by-cell basis across the entire tissue section. HeLa analyses can be done on the entire tissue section or defined areas of interest. So if you have specific regions of interest, RIs, we can only analyze those. Okay, so after image accusation, our next steps for HeLa analyses is identifying the region of interest, which is done by our HeLa experts, as I mentioned, in consultation with our pathologists. So we work as a team, and we believe that teamwork makes the dream work. So once the annotations are done, our Halo experts adjust the setting to detect the cells and each signal with high level of accuracy. So they pretty much analyze everything cell by cell. Okay, so now we're gonna go more in depth with viral detection, and we'll show you some case studies. And then for the second part of this part of the presentation, I'll hand it off to Jyoti for more in-depth analyses of transient viruses. So this is the slide you already saw at the beginning of this presentation. So this pretty much shows you the adeno-associated viruses, AAV, are proven to be effective with delivery vehicle with high transduction efficiency, stable trends, gene expression, broad tropism, and ability to achieve systemic delivery. However, this is essential to understand the biodistribution of the gene therapy and then in target and non-target tissues, as well as the persistence of transgene expression in those tissues too. So we use our RNA Scope assay to detect AAV transduced cells in monkey retina, as you can see in the top right corner. And then we also use I'm sorry. So yeah, so we use the AAV transduced detection in human liver, as you can see in the middle panel, and then we also can detect the AAV transduced signal in human brain, as well as some NHAP brains and etc. So for RNA Scope detection of AAV in homologous retina, we utilize RNA Scope to detect the vector driving GFP transgene expression under control of the CD promoter. And then this was injected subretinally into the eyes of homologous monkeys. So this RNA Scope probes were designed to target either CB promoter, which is the DNA sequence of the AAV vector, or GFP transgene mRNA. So in this slide, we see the overview of the retina probes simultaneously for the AAV vector shown in green, and then GFP transgene shown in red. So I say green, but I mean, we put this in more like a teal bluish color because that's how it appears on the as a final result, which we'll see in the next slide more in depth. And then GFP transgene is, you guys can see that in red as well. So we compare the naive region, which hasn't been transduced, and we compare it to a transduced AAV GFP region on the left side. And we can clearly see that there's no expression detected in the right side of the panel in any of the retinal layers listed on here. So if we go more in depth, we wanna ask the question, why is GFP signal so much more than AAV signal? This is because GFP is getting transcribed, expressed, and exported. The reason why CD promoter signal, you only observe it in the nucleus, so in the nuclei. One question that comes up is, are we detecting CB promoter RNA or DNA? So you can clearly see that there's a lot of saturation in the nucleus on the left side, and that's our CD promoter. And we know that from the basic molecular biology, we know the CD promoter doesn't get transcribed. So here the probe is antisense probe, we're detecting DNA, but because it's not integrated, so it doesn't require denaturation like integrated chromosomal. So another question that might come up is that in some cells, signals observed as blobs and in some as dots. So this cannot be controlled how much virus actually gets taken up So sometimes you see the variety or the spectrum of dot intensity or clusters. So now we're going to go through a couple of more case studies. And then going off of the first example, these are going to be pretty straightforward to understand. In this one, we only detect Cas9 transgene, which we can see in red, in the photoreceptor cells. And this one is detecting both DNA and RNA. And then it's supersaturated signal in singleplex Rna Scope Assay. Moving on, this is similar to the previous one where we detect AAV human codon optimized transgene in green, which we see as a cytoplasmic signal more so than vector genome, which is in red, and ciliary bodies of NHP eye tissue, and it's supersaturated in DNA. In DNA, I'm sorry, in the nuclei. And then one cool thing about this AAV application is that we can also pair up AAV DNA signal with one of the cell markers, which in this case is a neural cell marker, Faqs3 in NHP brains. And then this AAV vector is detected only in these these cell types. And we can also see the co-detection in the right image and the left image as well. So this is the way we can identify cell markers and where AAV is being detected. So AAV detection can also be used under our fluorescent platform, which you guys can see here on the left side of the panel, where we see the transgene being co-detected with our cell marker Faqs3 on the left side with no AAV treatment. You can only see the cell marker gene, and then on the right side in purple you can see the detection, plenty of detection of AAV treated sample. On the right side, this is pretty much showing the same thing in chromogenic duplex staining. And the bottom, you can, if you zoom out a little bit, probably around 5x, 10x magnification, you guys can see what type of, what part of the brain is being detected. And then AAV can also be analyzed with, and transgene, or it can also be analyzed with base scope. And then this gives more like granular and more punctuated dot kind of signal. So here we can compare vehicle treated or naive sample with the left side where it's AAV treated. And we can see a nice dual co-expression within several cells. And then we can also quantify base scope, AAV base scope as well, which you can see on the right side. So what we use to quantify, especially base scope AAV detection, is our Halo software from Indica Labs, which we have our whole imaging team, sub-team for this Halo analysis. Gives you granularly cell by cell analyses, as we've aforementioned. And then we have kind of like NHP, cardiac, and skeletal muscle, where we're targeting AAV vector and DNA. Only AAV vector, DNA detected in the liver. So this is only targeting DNA, and it shows a nice representation of this is RNA scope. This is not base scope, even though liver might look like base scope, but this is detecting the DNA signal in the nucleus for the most part. So aside from AAV vectors, we can also AAV treated samples. We can also detect our antiviral cells. And then for mRNA detection, we we hybridize the viral UTR region. And then for the DNA detection, we hybridize our probes with vector non-coding strand. So you can see right here on this little diagram. And then one example for SIV, simian immunodeficiency virus RNA, and polymerase DNA. You guys can see here how these are being co-detected. So our DNA signal is going to be in brown, and then probe for coding strand is going to be in red. And then the way these are analyzed is by doing a cell by cell analyses, which our Halo team does as well. Okay, so this is my time. Thank you guys for listening to this intro overview, and then we're gonna switch over to Jyoti for more in-depth viral presentation. Thank you.
Thank you, Mladen. Rhonda, can you hear me and can you see the slide properly?
Yes, we can. Thank you, Jyoti.
Excellent. Thank you, everyone, for joining the presentation today. And I think Mladen did a great job of just going over all the assay services offerings and showing you some of the beautiful work that they've done so far. So with keeping it in interest of time, I'm going to go over some of the COVID-19 offerings by ACD, and also show you a few publications that came out using our probes in the early COVID-19 research. So moving on, here are the product offerings for COVID-19 viral detection. As you can see, with the different platforms that are available using our technology and the different assays that are available, you can really look at the entire viral pathogenesis, the mechanistic action of the whole process. Starting with using our singleplex assays, you can detect the virus standalone, or you can, using our duplex or our multiplex formats, you can look at the virus receptor interaction. In addition, we all know that this COVID-19 infection has been associated with this cytokine storm. And so using our multiplex format, you can also look at the receptor immune response post-infection. And lastly, a lot of the tissues that are affected by this infection, we saw that researchers were studying the histopathological markers. So you can also visualize these pathological tissue markers as a response to the infection. So you can really cover the broad spectrum using our sensitive, specific single molecule detecting assay while giving you that morphological context. So a little bit more information on the actual probe. It's the VnCoV 2019 S probe, and it targets the coronavirus that caused the outbreak in China in 2019. Now, this probe is specifically designed to detect the S protein on this novel coronavirus. And that's really where the specificity of our probe design comes in, where it prevents cross-detection to the other species within that family, such as like the SARS from 2003, the MERS, or even some of the other viral species such as Ebola or HIV. And currently, we have probes that can be used in various animal models, such as the human, mouse, rat, monkey, even ferret and bat. And lastly, the most common way that this probe is being used is to study the localization and infiltration of the virus in tissues. But that's just the most common use. There are, of course, a whole plethora of uses that I'll show you some examples of in some of the publications. So based on the data that has been published since the emergence of this virus, ACD made available these RNA Scope probes to study the pathophysiology. So these probes have already been designed, and then you can use them to enable your viral detection research. So the first one, as I mentioned, the viral detection, right? You can look at probes to identify or detect the viral RNA on its own, But in addition, you can also use this sense probe to look at the replicating strand. And as you'll see in some of these publications, it is really important because you want to know the persistence of the virulence in infected tissue. In addition, the host cell markers that have been shown essential for the aid in the entry of the virus, and they facilitate the course of infection. So some of the receptors that we saw in literature, such as ACE2, TIMPRIS, and catepsin. So these probes are also available. In addition, like I mentioned, the cytokine storm. So inflammatory response markers that were shown to be infiltrated in the early times, they were shown in the lung tissue of patients with a severe form of the infection and patients that showed signs of acute respiratory disease syndrome or ARDS. And so this, using RNA Scope, you can really spatially visualize all of these immune markers, allowing you to identify that immune landscape at the site of infection. And lastly, as I mentioned, the histopathological markers, which can be used to study both the functionality and the homeostasis of tissues. These are some markers for lung, but of course, with increased research, there are markers that you can also use for some of the other tissues or organs affected by SARS-CoV-2 infection. So with that, I'd like to show you some examples of how the probes were used in the field based on some publications that came out. So the understanding of protective immunity to this SARS-CoV-2 is critical for vaccine and public health strategies aiming to end this global pandemic. What it really is basically, you read in the news, people are trying to understand that if a person infected with this virus, do they build an immunity to re-exposure? And so the key unanswered question is whether infection with SARS-CoV-2 results in protective immunity against this re-exposure. So in this paper, scientists from Harvard Medical School, they developed this rhesus macaque model for SARS-CoV-2 to study the pathological and the immunological findings of COVID-19. And in addition to that, they also use this model to study the natural immunity against this re-exposure to this infection. So as you can see on the lower right side, they use the RNA Scope chromogenic red assay to visualize both the SARS-CoV-2 viral RNA as well as its replicating intermediate in the lung tissue. And so this is really emphasizing on the fact that you can look both at the viral RNA as well as the replicating strand of the RNA in the same tissue context. In addition, now they also performed IHC staining for cytokeratin and viral nucleocapsid protein from Novus. Novus is a sister company of ACD, and this was done to indicate that the infection was in the alveolar epithelial cells. So to summarize, really, all this information from this paper can further be used for testing vaccines against the virus and also for development of therapeutics. So this is another paper that came out of University of North Carolina from the Barrick lab, in which, the mode of acquisition and causes for the variable clinical spectrum of this coronavirus disease remains unknown. As you know, that people out there that are infected, they all show different symptoms and signs. So really what causes that diversity or that variability is unknown. And so scientists in this study, they used a reverse genetic system to study the SARS-CoV-2 growth and pathogenesis. Now, they use both the chromogenic as well as the multiplex form, and they tested RNA Scope on cytospin samples. So that is once again showing the ability of RNA Scope assay in various sample types. And they revealed this ACE2 expression in upper respiratory tract with an increased sensitivity and efficacy, and they compared this characteristic feature to single-cell RNA sequencing. In addition, to the right side, you see they use the chromogenic red assay to establish an ACE2 expression gradient in the respiratory tract. and also to detect the TMPRSS2 in the normal human airway surface epithelium. So they looked at both the receptors as well as the viral expression. And we don't have the image of the viral expression on this slide, but you can go look up the paper. So now this is another paper that came out of US AMRIT, and this was one of those early papers. So what they were trying to do is they were evaluating various commercially available reagents, such as like antibodies, as well as molecular detection techniques to identify this SARS-CoV-2 in infected FFPE cell pellets. So they developed these cell pellets from African green monkey kidney cells. So they basically, assessed multiple IHC antibodies, and one of the technologies that they evaluated was Rna Scope assay. And so they saw the benefit of this assay. And so you see on the top image, they've used the chromogenic red assay, once again, to detect both the spike protein as well as the replicating intermediate in these FFPE cell pellets, and also the multiplex fluorescent V2 assay. So they show basically use of both these platforms. But what you really see here is the specificity of the assay, which you can see when you compare the data from the uninfected to the infected tissue with little to no background in the uninfected tissue. In addition to that, now they also perform dualish IHC. It's the technique that Mladen talked about before that allows you to simultaneously detect mRNA and protein on the same tissue context. Interestingly, they actually looked at both the SARS-CoV-2 mRNA and the protein on the same FFPE pellet. Really, once again, highlighting the power of using RNA Scope, not only standalone, but also the ability to look at RNA Scope with a protein on the same morphological space. And this is another image that is a paper that is coming out of Yale School of Medicine. What they wanted to do is this is a case of a COVID-19 positive patient, and she was in her second trimester that was complicated by severe preeclampsia and placental abruption. So the researchers at Yale Medicine, they wanted to distinguish the effects of preeclampsia and COVID-19 so they could better prognose the pregnancy. And so they used the chromogenic brown assay in this case. And as you can see, they visualized the SARS-CoV-2 in the placental tissue. They also compared this to the ACE2 receptor expression in the human protein atlas, and they found that the staining pattern closely matched that of ACE2 in the HPA. And lastly, this was one of those other early papers. This paper, actually, they started working on this in 2019, and it came out of Chinese Academy of Medical Sciences. So at that time, plenty of diagnostic, preventative, and therapeutic knowledge of this virus was enriched, at least in China, from clinical studies in 2019 in December. However, the animal models, particularly non-human primates, were urgently needed because researchers could not answer certain critical questions in clinical patients, such as evaluations of antiviral drugs or vaccines. And so the need of non-human primate species was urgent. So these researchers, they evaluated various families of non-human primates to identify an accurate model to study the infection. And they found out that the M. mulatta was more susceptible to the infection compared to the M. fascicularis and the C. jacus. So what they used is they used the RNA Scope fluorescent assay to basically look at the viral distribution in different tissues, really demonstrating the high sensitivity and specificity for the SARS-CoV-2 detection. So with all these examples, I still wanted to show you a couple of examples outside the COVID field, right? I mean, the applications for RNA Scope can be applied to all viral species. So here's an example from Erasmus Medical Center on the MERS-CoV. As you know, MERS falls under that family of coronaviruses. And so at that time of the outbreak for MERS-CoV, it was known that the transfer to humans was fueled by introduction of this virus from dromedary camels. So scientists from the Albert Osterhaus group, they showed a viral vaccine, the MVAS, that apparently confer mucosal immunity to the virus in these dromedary camels. So as you can see on the top right image, they compared using RNA Scope the viral expression in a control group versus the vaccinated group. And they saw a significant reduction of the viral expression in the vaccinated group. In addition, they also compared the mRNA and the antigen expression on serial sections using ISH and IHC. Now, this was an old paper when MERS had happened. So if this was a recent time, you could use, again, the dual ISH-IHC workflow that Mladen talked about to look at this data on single tissue and in the same tissue context. Another example that I want to show you, and this is one of the last papers we'll look at in today's presentation, is this is an example of Zika virus. So when Zika virus outbreak had happened, it was causally linked to congenital microcephaly and also in some cases to pregnancy loss. And so to understand its intrauterine transmission, replication, its tropism, and once again, the persistence of this virus, that is often not studied in detail with some of the other techniques was really important. So in this paper from CDC, Bhatnagar and his researchers, they used the Rna Scope chromogenic assay to localize both the replicating strand as well as the viral RNA in brains of infants and placentas of women with pregnancy loss, demonstrating that the viral infection replicates and persists in fetal brains, further showing its association with congenital microcephaly. So we've really seen that broad application from not only viral detection, but also vaccine development and also therapeutic strategies. So with all of that, publications are so important. And so the growth and adoption of this technology is best exemplified by the number of peer-reviewed publications. Our first publication was in 2011. And since then, there have been over 2,700 peer-reviewed papers published using the RNA Scope technology in numerous journals, and many of them are in top-tier journals such as Nature, Cell, and Science. The technology is highly relevant across multiple fields of research, with over 40% of these publications being in cancer, infectious, and of course, since 2019 and 2020, we've seen a significant growth also in infectious diseases, but also in other areas such as stem cells. Development has taken a lot of growth, inflammation and such. And so the publications are really showing you the power of this technology in research out there. So with all that Mladen is covered today, and I've shown you, I hope we've convinced you today that the RNA Scope uses a unique probe design strategy to dramatically improve the signal-to-noise ratio. We have over 22,000 probes on catalog, but our rapid probe design allows you to design and make probes within 2 weeks for any targets in any species. Some of the examples I showed you earlier, to make an antibody for some of these new species may take longer. So in case of infectious diseases where you need results faster and you need assays faster, this assay really helps. RNA Scope provides both quantitative and spatial information on your targets in that complex tissue environment, but it's really providing you all that at that single cell resolution. And you can multiplex up to 4 targets simultaneously on the same tissue context. Our newest base scope assay can detect splicing variants and short sequences down to 50 nucleotides. And also the RNA Scope, as I showed you, can be complemented with IHC or IF to look at both RNA and protein on the same slide simultaneously. And all of these features that you can see here, all those applications that you can see, they can be enabled at especially such unprecedented times using our assay services. The team does a great job, and you can see some of the success stories or the accolades for our teams that they've received for their beautiful staining, their reproducibility, their ability to support you on your projects from the start to the end, and just like the quality of the data that you get out of the assay services work. So with that, the last thing is our FAQs. So if you have any questions about anything that you studied today, feel free to enter it in our Q&A box directed to all panelists. And our support team and our panelists will reply back to you.
Great. Thank you so much, Jyoti and Mladen. What excellent presentations we had from both of you today. Just a reminder for the audience, if you do have any questions, we are hanging on for a few more minutes to take questions in our Q&A box. Also, this webinar has been recorded, and so look, for the next 24 hours, we will post this online at our acdbio.com website under the recorded webinars section. So again, once again, thank you very much for joining us today. We hope you learned a lot.